Hey gang! So in this unit, we're going to talk a lot about genes. And so before we do that, I want to talk about what that means. So gene is really just a short way of saying a piece of genetic material. But what exactly is genetic material? So when we talk about that, we're talking about the molecules that store information for determining how an organism is created and maintained. So that's the stuff, the molecules in your cells that are basically giving out all the commands for how the cell and all the neighboring cells and the whole organism, basically, which is just made of cells, how it's made and how it's maintained, how it continues to grow and develop. So the genetic material has the information for, or basically controls, all the traits that we can see in an organism. So the way it looks, what it does, all of that is determined by this information stored in the genetic material. Genetic material is present in every cell. And so it needs to be tough enough to last for the life of the cell. You wouldn't want genetic material to be made out of something that can break down really easily or maybe get damaged super easily. So it needs to be some tough stuff. It also needs to be copied and passed on to new cells exactly so that they have exactly the same information. When one cell divides to make two cells, you want those new cells to have the same traits as the old ones, so they need to have exact copies of the genetic material. So these are kind of the three main things that genetic material needs to be able to do. And we know today that the genetic material in our bodies is DNA. DNA is the molecule in all of our cells that stores the information for our traits. But back a long time ago, they didn't know that DNA is what did this. They didn't know what molecule it was. So in this lecture, we're going to talk a bit about a couple of experiments that helped us find out that DNA was the genetic material that gives us our traits. So first off, we're going to do a little bit of background stuff so you can understand these experiments, and they involve bacteria. So take a second and think, what do you know about bacteria? Well, let's go through about some things that we've learned already in class about bacteria. So first off, they're alive. Bacteria are living organisms. So that means there are 8 different things that we know they can do, right? That we talked about in the very beginning unit of what makes something be alive. So one of those things was metabolism, or being able to take in food and break it down and use it for chemical reactions and stuff. So being able to take in and use energy. Also the ability to replicate, right? Bacteria can make more copies of themselves. And they can do all those other things that we talked about that living things can do. Bacteria are always single-celled organisms. You might find them hanging out in clumps together like this, but there's just one single unit. This is one bacteria, this is one bacteria, this is one bacteria. They never grow more than one cell that works together. We also know that they have DNA. That's one of those things about being alive that you have to have. But since bacteria are prokaryotes, that means they don't keep their DNA in a nucleus. They don't have any special organelles like a nucleus inside. They're pretty simple. And also, just a side note, their DNA is usually circular. So they'll have a little piece of circular DNA so that it's one molecule and it's kind of connected in a circle there. And then something you probably already know is that some bacteria are bad for you and they can make you sick. But some bacteria aren't bad. They might not do anything to you, and some bacteria are actually really useful. So we have a lot of bacteria that lives in our gut that helps break down food for us, and we would be in pretty bad shape without them. So some bacteria we do need, and they also make useful things for us, like yogurt. Yogurt has a lot of bacteria, so we like them. And then one last thing that we know is that they have different traits. So for instance, this green bacteria, it's a rod shape. That's what this shape is called. This one's obviously round shape. And then there's some that are kind of a spirally shape like this. They might be different colors. They might be able to live in different places and eat different things. Some have the ability to move and maybe some don't. They all have different coverings on the outside, so they all have a cell membrane, but some of them have a protein coat around that, or they might have different proteins built into their membrane. So all these different traits that they have, each bacteria, or each type of bacteria, I should say, is unique and different. It has different traits, and that's caused by the different proteins that they have in their single cell. Now, a really cool thing that bacteria can do that we can't is a process called transformation. And that is a really cool process of gaining new traits. So bacteria always take in food and nutrients and all the things that they need from their environment through the process of endocytosis. And if you remember, that's basically just wrapping their cell membrane around something and bringing it in. So besides just bringing in food, they can also take in genetic material from their environment. Now this would be available if, say, this bacteria and this bacteria were hanging out in a pond together, and this one dies and it breaks apart, and now it's got proteins and lipids and all those molecules it was made out of are now just floating around. So some of its bacterial DNA, right, this genetic material, is going to break apart into bits and pieces, and this bacteria can take those in, and instead of digesting it and using it for food, they will incorporate it into their own DNA. So all of these bits of genetic material, they are the information for making new traits, basically, right? So once this bacteria takes in this new genetic material, it's going to have new traits. So this bacteria is going to have now some of the traits of this old one. So that's a pretty cool thing that bacteria can do that us big multicellular organisms can't. So now that we know that, we can dive into this experiment by Frederick Griffiths. And he took some bacteria. It's kind of a pneumonia bacteria. So this one, it had a smooth outer coat, so he called it the S strain. And if he put that into a mouse, the mouse would die. So this was pretty lethal bacteria. And after the mouse died, they tested the blood, and yep, sure enough, there are these S strain cells living in the mouse. Now, this bacteria had a relative called R strain because it had a rough coat, R for rough. And this bacteria was pretty harmless. If you inject that into a mouse, then the mouse would live. And then after a while, if you tested its blood, you wouldn't find any of these bacteria in it because the mouse's immune system was able to fight them off. So his experiment involved heating up these bad S strain cells, so basically killing them. And then he took these dead S strain cells and put them in the mouse to see if they would still kill it, and no, they didn't. So the mouse stayed healthy. He was alive. Tested his blood later, didn't find any bacteria, as expected, because it's dead. But the really interesting thing happened when Griffiths mixed this nice R strain bacteria, which didn't make the mouse sick, with the dead S strain cells. With those two mixed together, two things that wouldn't kill a mouse, right? The R strain by itself didn't kill the mouse. The dead S strain cells by themselves didn't kill the mouse. But when they were mixed together, the mouse died. So Griffiths figured out from this that these nice little R strain blue guys that normally didn't have what it took to kill the mouse, they were taking in genetic material from the dead S strain cells and incorporating it and using it, and they became themselves more like an S strain cell. So when that mouse died and they tested the blood, sure enough, they found live S strain cells living in the mouse. So mixing these together formed a new cell that looked a lot like this one, because these R strain cells took on the traits of these bad S strain cells, and those traits that they now expressed killed the poor little mouse. So that was really interesting, finding out that genetic material can be transferred from one bacteria to the other, but it didn't really tell us anything about what that genetic material was made of. So somebody else took this experiment a bit further, and his name was Avery. And he took some of these heat-killed S strain cells, the same kind that Griffiths used. So here's showing them dead. He mixed them up, and he figured lipids do not have what it takes to be genetic material, and neither do sugars, because they're both very simple molecules, easy to break down. He knew they couldn't possibly be what was coding for all of our information, so he just took those out. What's left, the only other molecules that are left in something, would be proteins and then nucleic acids, both DNA and RNA. So to figure out which one of those things was the genetic material, what was passing on the traits, he split this into 3 different tubes. In this one, he used something called proteinase that would kill all the protein, break it all apart, because that's an enzyme that breaks down protein. So if all the protein gets broken apart, could those R cells take up genetic material and become S cells? Yes, they could. That means it wasn't protein that was passing on the traits. Did the same thing in this tube, added RNase, an enzyme that breaks down RNA. So now there's no RNA left in here. Mixes it together with the R strain cells, and yep, S cells now appear. Those R cells could still take in that genetic material, so RNA wasn't the genetic material. So then in the third tube, he added DNase, an enzyme that breaks down DNA. So now in this tube, there's no more DNA. We just have the proteins and the RNA left in here. Mixes it with the R cells, and voila! No S cells appeared. That means the R cells could not take in the genetic material. And remember, the only thing that they got rid of here was the DNA. So that meant DNA was the genetic material. DNA is the molecule that was passing on traits that the R cells could pick up and use. Without DNA, those R cells could not gain new traits. So this was really a breakthrough experiment that taught everyone that DNA is the genetic material. That's what has all the information that's needed to give you your traits. So take a minute and fill out your worksheet. And if there's any part of this that you didn't understand, then definitely raise your hand or find a teacher and we'll help you out. See you later. Wait, one last thing I forgot. Okay, I had one more slide. So the DNA is our genetic material. DNA codes for the proteins. And remember, proteins are what cause all those traits you can see that make up our cells, that make up our bodies. All right, goodbye.